Okay, just a very brief recap from yesterday. What we talked about is transformer models. You have seen how they are used in language, and you have seen how they are used for proteins sequences. So we have seen how you have to basically change your sequence into from letters to numbers, basically. So this process is called tokenization, and we've seen how these transformers work through the attention mechanism. Okay, so the attention mechanism allows these models to learn from the context that they see. So this is very important because they can learn long-range correlations between your amino acids in the sequence. And we've seen that they can learn without being supervised in any way, so you don't tell the model anything about the structure or the function of your protein. They can learn the contact map just by looking at sequences. Okay? And we have seen also a model that is called, it's kind of important, it's called the MSA transformer. Remember what an MSA is? It's a multiple sequence alignment. This is very powerful to predict contacts, to predict the structure, because it uses the information about coevolution in your homologous sequences directly. And this helps. So now in this lesson, what I will do is to show you some applications. So how these protein language models are used, and yeah. So mainly, we've seen that these models work as encoders. So they make a representation in high dimension of your sequence. The way they are typically used is with what is called transfer learning. So the exercise from yesterday was exactly this. You take these representations and you solve some downstream task. This is called downstream task because the training of these models is called pre-training. So they are trained on like very large number of sequences. And then in the end, they learn these very meaningful encodings that can be used to solve tasks that you typically, before they exist, you were solving with sequences. Basically, you started from sequences and you did, for example, classification into alpha or beta structured proteins. We started from embeddings, and the performances improved, or should improve, in principle. This is how they are used. And so, the,
我有好方向了。
no, it's fine, I can do that. You can do
或者没有我控制能打过。
that. Okay, that's fine. So, protein language model just these sequences. And they learn from evolution. Evolution works by basically accumulation of mutations in the protein sequences. And these mutations can be of different type. Can be neutral mutations, which means that they don't affect the protein structure or the protein function. And this can happen if you remember the amino acids classes. You can have a substitution that is within a class. So, for example, from hydrophobic to hydrophobic, this is very likely not to affect the protein structure and function. And so you'll have a neutral mutation. But, so why mutations are important? Neutral mutations are important because they give you basically homologs, right? So proteins that, remember, fold in the same three-dimensional structure. But you have even single point, what are called single point mutations, which means just one amino acid is changing in your protein sequence. Even with one of these, your protein structure could be disrupted. If this mutation occurs at a particular position of your sequence that might participate in a contact, then you could have a disruptive effect. So your protein either doesn't fold anymore or it folds wrongly. Okay? These are very much related to diseases, of course, because if your protein functions is lost, you might have some sort of disease. So this is why it's important to predict them, to understand what mutation could be disruptive, could help therapeutics, for example. Okay? And then there are favorable mutations. What are them? They are basically mutations that improve the stability of your structure. So your structure is formed because there is physics laws, right? So the amino acids are molecules, are made of atoms, and they make interactions. So in the end, you obtain this structure, there are some stability, and you could have mutation that improve this stability. These are favorable. Why these are important to predict? Well, it's kind of like trying to understand where evolution is going, so it's cool, per se, let's say. But also, if you see it, for example, from the point of view of a virus, these are also very important to predict because viruses, they mutate in order to infect cells. They live when they have a host. They don't live, let's say, alone. So it's really important to them to be compatible with your cells. So what happens is that, for example, if you remember COVID pandemics, we, at least in Europe, we had to vaccinate. I don't know how the situation was here, if it was like you had to vaccinate, but in Italy it was like that. So we had to vaccinate, and then at some point they told us, well, you have to vaccinate again. We were like, what? We just vaccinated. What do you want from me? So the point there was that basically the virus, okay, so there is one thing to say, evolution in like living systems, let's say, it's kind of slow compared to viral evolutions. They mutate much faster because it's their strategy to adapt. So they mutate very, very fast. And what happened is that if you vaccinate, you're inducing a response of your immune system to recognize, basically through your antibodies, to recognize what is infecting you. So how did this vaccine work? It worked exactly relying on spike protein of COVID-19. What is the spike protein? It's the protein that is at the surface of the virus. So if you have in mind, now I don't have it here, sorry, but if you have in mind the typical cartoon that one uses to show a virus, this is like a ball with some spikes on it. Yeah, those are the spikes, exactly. So those are the proteins, these are proteins. They basically are the proteins that attach to some receptors in your cell. I've seen that Jacopo showed you what a receptor is, more or less, if you remember the membrane of the cell with this protein in it. This is a receptor. So these are proteins that are used by the cells to basically communicate with the external environment. So stuff can pass through these receptors and get in or get out. So one of these receptors is the one that is used by spike to infect you, basically. Well, the spike will interact with your receptor, so it's a protein-protein interaction. And when this happens, the viral material enters your cell. So if you train your immune system to recognize this protein, at some point you will be immune. You activate your immune response. So the way these vaccines were made was exactly by inducing the production of the spike in your body, so that the next time that COVID infects you, basically you will respond very fast to it and recognize it. So at some point, we basically created a barrier for the virus. And so what the virus wants to do is to escape this barrier. And this slide is about this viral escape. So what happened in reality is that the spike protein mutated. Mutated in such a way that our antibodies cannot recognize it anymore. So when this happened, we need to revaccinate again, because the spike changed, and so the vaccine changed. So of course, from the point of view of the virus, this is a positive mutation. It helps it basically functioning. And I hope you get the feeling of why it is important to predict them. So protein language models are really great in predicting mutations. And so people started to use them to do so. So if you remember, at the end you get, from a protein language model, you get naturally, because of the loss, which is, remember how it works. You mask some amino acids and you ask the network to reconstruct their identity. So in the end, you naturally get some probability for amino acids to be there. And you can directly use these probabilities to build a notion of like what mutations are closer to me. So if I'm a sequence, I will have a certain probability of having some amino acids there. And so you can go to the one that is more, a little bit less likely than me, but still likely to be there. So you can build a graph with your protein language model vectors. And now you have like a notion of proximity of your representations. So you basically take the representations that are the closer to each other. Okay? With the Euclidean distance, you have these vectors, you calculate the neighborhood of each representation, and you build what is called a KNN network, K nearest neighbors network. So you fix how big is the neighborhood that you want. So how many close sequences you want to consider. And from this you obtain a graph. You see, this is a sketch of a graph. You see that there are errors here. So the direction of the errors, is given by basically the probability of observing that amino acid there. So if this probability, the difference, so suppose I'm a sequence here, okay? I will have some letter in some position, and I compare, and that letter will have a certain probability for the model. Then I compare with the one that is more likely to be there. So I look at probability differences for each amino acid. And this gives me the errors. So where the basically most favorable thing is going, the most favorable mutation is going. So you obtain a directed graph in this way. And there are statistical methods that allows you can see this as a diffusion process, whatever. So you can solve the graph in some sense to find the roots of the graph. So you can ask yourself, where did this origin from? So it's like a simple game to play. It's not that difficult. And when you do so, in this work, they did it for several viral proteins. And this one is nucleoprotein from influenza. Why influenza? Because we have a lot of data from influenza. Simply because of that. So you remember that you can project in 2 dimensions your objects, basically your high dimensional objects. So you can just plot them, basically. And what you see here is that when they play this game with this protein, basically you see, so this is the color is like the real time in history in which that mutation occurred. So you see it goes from 1918 to 2020 here. So they have very many data. And so the representation is from the graph you obtained from the protein language model representations neighborhoods. This is like having solved the roots. So the black dots here basically are the roots that they found from these graphs. And it's very interesting that these roots correspond to pandemic events. So it's like they are really the root for your influenza protein evolution process. They are like important events from which a pandemic started. This is like a possible application. I think there is also, yeah, I also show you here the COVID-19 spike situation, basically. So of course, here the real time it's much shorter. It goes from 2020 to 2021. And yeah, basically, when you play this game, you see that the pseudo time that is obtained from your graph, solving your graph roots, it's not like that. It's called pseudo time because it's not a real time, let's say. It's something that you model. But you see it's pretty much corresponding to the real one. So stuff that came, mutation that came with the word before are actually reoccurring when you compare it with the real time in which they. So this is nice. It gives you the idea that you can basically predict stuff in the future. Because if you know the evolutionary trajectory of these objects, most likely you can hope to predict something that will be next, which for viruses is very important because, of course, we want to know in advance what the evolutionary pressure is doing on the virus sequence. For example, for vaccine development. Okay. Then I want to talk to you about this model. It's a little bit a change of topic. So remember that protein language models are called, there is a big class of models that is called ESM. And ESM3 is like the most recent model that came out, and it has some particular feature, which is multimodal. So if you see here, that's the input of your model. You have sequence, but now you have also structure and functions. So you can, it's nice because it's a multimodal in the sense that these 3 types of data, they are completely different, right? Because the structure is a set of positions for your in 3-dimensional space for your atom. The sequence is just a sequence of amino acids. And the function is like text annotation. It's the function of the protein, but it's expressed as a sentence, basically, in these databases where you have annotated this function. So these are very different objects. And the nice thing is that you can tokenize, as we've seen, each one of these objects and make it, let's say, live in vectors that have all the same dimensions. And when you play this game, you can just fuse these vectors together at the beginning and then pass it to the network. The network is a transformer model. But it has one new thing, one different thing, that is the geometric attention. Geometric attention, we will look at it, is just to be able to deal with structures. Because the structure needs to be rotational and translational invariant. And so what you did with the structure, it's a good question to ask, like how do you tokenize a structure, for example? And what's done is, in simple words, it's like for each amino acid of your sequence, you have a structure, because of course this is strained structure, so you need to have it. You can look at the structural neighborhood of that amino acid and map it to a vocabulary. So you will have, imagine you are an amino acid in a structure, you will have some neighborhood, right? Some amino acids that are close to you, that could be far away in the sequence, by the way. And so you have like a local structural pattern. And this can be seen as an element of a vocabulary, just as like the vocabulary of your amino acids. And this vocabulary size, in this case, is for more than 4,000. So it's pretty big because, of course, you have many, many ways in which these amino acids can arrange in structure. Okay. So one last technical thing. We have seen how these language models are trained with what is called basket loss. And this is what also this model is doing. But the problem, well, not the problem, they found a nice solution to make this model generative. So it's not just predicting something. This model can predict a structure. This model can predict a function, for example, because it has these modalities in input, so it has to have it also in output. You can use a decoder and obtain and go back to obtain a sequence, a structure, or a function. But it can also be generative, so you can basically tell the model just to generate a structure or a sequence with a given function. Just by saying, for example, I want these amino acids in this position and this other amino acid in this position. This is a prompt that you give. It's like a suggestion that you're giving. The model will generate a sequence with that constraint. And this is done basically by not masking just some random amino acids that we've seen yesterday in sequence, but playing this game many times and masking each time you run it, you mask a different fraction of amino acid. In this way, you obtain a probability distribution, and you can sample from this probability distribution. This is how this model is generated. And the cool thing is that they showed, okay, so the guys that invented this model, they founded a startup. And so of course they want to sell it, but I think they sell it in a very nice way, in the sense that they show that these green fluorescent proteins, okay, green fluorescent protein, they emit light, this way. And are very much studied. There are many sequences and structures that you can look at. But what they did is like just, so they have basically this fluorescent spot that is called chromophore, which is the one with the light in the picture, in this cool picture. So they just tell the model, okay, this is the position of a chromophore. Design, generate a sequence and a structure with these constraints. They play the game, and what they find is that basically they generate a protein that is a green fluorescent protein, nice structure. It is exactly the protein that they want, but apparently it is very far from all in sequence from all the sequences of the green fluorescent proteins that are known. And they claim, maybe this is a bit overclaiming, also because it's kind of hard to measure this, that they're like, this protein that they generate is like 500 million years of evolution away from the ones that we know. So it's like, it's a way to say that they generated a complete new thing. It's like what protein design is in the end. It's kind of hard to assess that your protein, well, for sure you can say that this particular sequence doesn't exist in your databases. It's kind of hard to say that it's far in evolution from what you have, but they claim this. So, okay, we believe them. Okay, and now I think this is the last application that I want to show you. Oh no, but I skipped a very important slide. Ah, no, it's not synchronized, sorry. Okay, I told you that there was this Nobel Prize in Chemistry last year. So there are three guys. One is David Baker, which is very famous guy that was working with machine learning for a long time, and is particularly focused on protein design, which is exactly the thing that I just showed you with the very interesting protein. But then there are these two guys that are actually, they are from Google, DeepMind. So it's kind of also impressive that DeepMind has a Nobel Prize. It makes you think, let's say. They developed a model that is really, really important, that is AlphaFold 2. So AlphaFold 2 is a model for predicting the protein structure at the atomic level from the sequence alone. And yeah, so I want to show you how this works in a, okay, so see it's not really, we don't go too much deep into this, but the important part is that this uses transformers. This uses protein language model. It's a complex model. It's modular. You see there's some blocks. It's not just like a nice transformer model that goes from input to output. It combines stuff. But this stuff, in the end, is protein language model. And one important thing that I want to point out here, so how does it work? So you pass it a sequence, and the first thing that AlphaFold is going to do is to search for homologs and build the alignment. This is something that is very important for all I said, I think, up to here, because it tells you that this information is important. It tells you that evolutionary information about the protein is used by this model. And people tend to underestimate this because what you see, you can run AlphaFold online, you just pass it a sequence. So you don't see this step, but this step is really crucial. It's the first thing to do. Then so importantly, AlphaFold is a very good model. It wouldn't work if it wasn't supervised with protein structures. So the guys took all the protein structures that we know in the PDB and use it to train this model. In the end, when you make your prediction, you make it on a sequence that you don't know the structure of course. We have seen that there are many of these, that there are many sequences that we don't know the structure of. So, but to train, they use the structures. So basically what they did is to tokenize the structure, kind of like I showed you before with the neighborhood of an amino acid. In reality here, they put like relative distance between amino acids in the structures. They play a slightly different game. But in the end, it's like a structural tokenization. So you have these two objects, and now what you do? Transformers. So the one on top is the MSA transformer I showed you yesterday. Remember, it was working like a transformer, so you do playing the attention mechanism between amino acids, evaluating these correlations between amino acids in a sequence. The MSA transformer adds also an attention that is between sequences in an alignment. So for a given position, it will tell you basically the attention, the vertical attention in your alignment. So this gives you a representation for your sequence, while if you make just a single sequence transformer out of this structural optimization, you have again a representation for your sequence. And what this evoformer plot does, which is crucial for output, is to combine the two. So basically, it runs the MSA transformer, it runs what is called let's say the pair transformer, which is on the structure. And at some point, they will communicate. So the two representations are matched. Okay, then you will end up with a very rich of information representation for your sequence that is passed to the structure module. The structure module, I don't want to go too much into the detail because in the end, it just, let's say, extract information from these representations from the error format. So, yeah, it's important because it allows you to map these high-dimensional vectors on a structure. So you go back to three-dimensional structure with all the atom positions. So of course it's important, but let's say that all the information is already here. So what the structure model does is basically to impose this rotational and translational imbalance. It rotates the tokenized structure that you obtain, and yeah, it basically predicts angles, the torsion angles that we talked about in the very first lesson. So it gives you the protein backbone. Those torsion angles, if you remember, were in the protein backbone, which is the constant part. It predicts the atoms of the position of the protein backbones, and then it fits the real amino acid that you have, because it knows it, because it's in the sequence. Yeah? It's not working. Okay. Then there is a very important thing in AlphaFold, which is it's not just predicting very nicely the structures, very nicely, very accurately, but also it has a way, a score that tells you how confident the model is in the prediction, which I think is very important because sometimes the model can make mistakes. And so there is like an extra network, simple network, that is trained on two structures. Then basically what it does, it tells you how wrong you were. So with a true PDB, with a true structure, and you're predicting. with the structure. It tells you how close they are. So this PLDPT is a percentage, it goes from 0 to 100. And the nice thing is that when it's below 50, it's really a bad prediction, and it really is. So the model will always give you a structure in output, but it will also tell you, look, this structure is crap, or this structure I'm really confident in it. And this is useful. It's much related also to flexible regions of the protein. So when you have a flexible regions, the position of the atoms can be wherever. And so the PLDPT, which is local for each amino acid, will tell you, okay, here I'm low. But that's because it doesn't know where to place your atom, because your atom can be everywhere in the flexible region. Let's see if I have. Yeah, it's color coded. So for now, for example, here, your structure is all blue. It's of course, it's like a convention that they use. So blue means high confidence. Here I don't have an example of a low confidence one, but you see it red. So it's really visual. It will color your structure with the confidence. Okay. So AlphaFold is great, but sometimes the confidence can be really, really low. Can you think of a case in which this confidence, the prediction is crap from what you know? Like, for example, I told you this object is important, no?
What's that?
The multiple sequence alignment.
Yeah.
The usual multiple sequence alignment.
If there is no alignment or something like that.
Yes, that's it. So, for example, viral proteins, they tend not to have such a deep alignment because they mutate so fast and they depart from everything else that we know in evolution. So when you look for homologous sequences, you don't find many. And AlphaFold is not going to work well in that case. Typically, that's what happens. There are other cases. There are particular types of proteins that are called disordered proteins because they are not so much structured, they're very flexible. They are important also in the brain, and yeah, because they play a role also in diseases of the brain. But the point is that there it's very hard for AlphaFold to tell you where the atoms are because the protein is so flexible, it doesn't have a regular structure like common proteins. But this you couldn't know. The key point is the alignment. And that's why I stressed this a lot during the course. Yeah, here just to say that what the guys did, well, they wrote a Nature paper, then they got a Nobel Prize for it, and then what they did is like a service for the people, which is running AlphaFold on all the sequences, the Uniprot sequences. Remember, Uniprot is the database with all the sequences that we have sequenced, so all the sequences in the world, let's say. They run it for each of them, and you can just go in this database. You have an Uniprot ID which identifies your sequence. You pass it here, you get a prediction with the confidence scores and all the nice things, statistics that you can get. Okay. So with this, like, okay, one could think, since AlphaFold solves the problem structure, the structure prediction problem very well, one can think that this is like over, that structural biology is done, basically, with this. This is not the case because there are some challenges that I think they are very open research lines that we can investigate. And one is about interactions. So AlphaFold 2 is given, it will give you just one single structure. It doesn't deal with, if I have two proteins and then I want to know how they interact, it doesn't deal with it. But of course, the guys partially solved this because it's, let's say, you can do it kind of easily from AlphaFold 2 to extend AlphaFold 2 to deal with two proteins, basically. It's not just, and they did it through a model that's called AlphaFold 3. It's a little bit different from AlphaFold 2, but the key concepts are still the same. And this AlphaFold 3 also deals with other type of molecules. So it's nice because I mean, there are not only proteins, there are also RNAs, and there is DNA. So with AlphaFold 3, you can see the interaction between a protein and an RNA molecule, for example. It makes this prediction. It's not as good, I would say, as AlphaFold 2 in predicting these interactions, but it's okay, let's say. But there's still some research to be done there, and let's say there is room for improvement. On the other hand, if you go back to the mutation thing, you want to know the effect of a mutation on a protein. That's very important. We know why now. AlphaFold 2 is not able to do that. And in general, it's very hard to do that. Why? Because it's, even though protein language models are really much suited for it, the problem is that in machine learning, you need data to train your model. And let's say in the case of mutation, you might want to have some ground truth on some mutations that are bad, some mutations that are good. You want to know to train your model. So these data are kind of hard to get. And that's why this is a very open problem. What these mutations could do to your protein, it's hard to know. For sure, AlphaFold cannot do it. You take your sequence, you predict your structure, then you mutate one amino acid, you substitute it with another one, it's not going to give you the right structural effect. It was not trained for that, basically. And one last point is dynamics. So we've seen in the first lesson that, okay, we have energy landscape, we have physics, right? So these proteins are, let's say, undergo a force field, and they fold. They fold, minimizing their energy, but there is just not one minima to remember, one sharp minima. You can have different stable states, so different structure that the protein can have. There are some like very extreme cases of this, like for example, there are what are called fold switching proteins. These are very extreme cases because they are proteins, for example, circadian proteins. They work with the life, with the cycle, day and night cycle of your cell. So they do something at night and something completely different at the day. They exist. And when I say do something completely different, I mean they assume a completely different structure, the same protein. So you don't have one static frozen structure many times. You have a conformational landscape. You have ensemble of structures that are functional. So they are needed for do some function. So yeah, and also the way, which is tightly related to this, the way a protein moves can be important. So the dynamics. So nowadays we don't have good AI models to deal with this problem at all. I would say the problem is still with data there because there are actually no experiments. Well, there is a type of experiment that is like temporal X-ray. So if you remember the X-ray experiments, which assess the structure, you can do a very fancy and cool experiment with the X-ray, which it's gonna basically gives you the structure in time. So it's kind of like following the dynamics. But this is not so common. It's not so easy to do. I think there is one group in Poland that is doing that, and I don't know, few others. So basically, you don't have experiments to assess the dynamics.
So there are like atomic models, right? But they are like too short, like time.
You mean AI or you mean molecular dynamics?
No molecular dynamics.
Yeah. I was gonna say.
Ah,
so-
sorry.
No. Nice catch. So the point is, but these are in silico, so are simulations. They are made in computers. So we can kind of trust them for proteins because the force fits are really, really good. So remember, I mentioned molecular dynamics in one slide in the first lesson. So you have these classical approaches in which you basically define a force fit for your protein and you solve the Newton equation of motion. Basically, these are very tough simulations, very expensive because you have to track all atoms of your protein. And but for proteins, these force fields are really, really good nowadays. The problem is that you have to wait forever, etc. So what's the problem with? And also, someone would say, well, these are not real data; these are still simulations. So, but I mean, good enough. What I want to do in the next future is to work with this problem. So, but to do that, I need data, basically. So I would go a little bit beyond the fact that these are not experiments. I mean, we work with what we have. These are good simulations. We can trust them. We assume they are like experiments, let's say. Okay, good with me. The problem is that how many of these simulations do we have available in databases that are curated, that are open, that you can, like the ones that I showed you for sequences, for structures. Do we have something like this? Currently, no. Well, the right answer is that people is starting to collect these trajectories. These are trajectories, no? For like the movement of your protein. And yeah, so hopefully we will get there. But how this went until now is that people were just not publishing the data. So I want to make a point here. Maybe I think this is important because with the AI and with the importance of that we're giving to AI nowadays, because we realize it's powerful, it comes a discussion on data. These models are trained on big data, so you want high quality data, you want a lot of data, and people are like very much worried now about, also a little bit worried about AI, let's say. And one of the concerns or the criticism is exactly about data. Maybe like we want everything open. We want an open code. By open I mean that you can have it, read it. It's not private. So they should release. We should all release what we do. And I think this is not just about AI. It's a general philosophy. But so okay, it's okay to be critical, but I think we are improving in this sense because kind of like AI is forcing us to share the data also in like a correct way. But people were not doing that before. This is where I wanted to go. So, for example, to obtain this trajectory result. And for me, this closes this course. I don't know if you want to talk about something, you have questions, you have observations. Yeah,
Have you worked in molecular dynamics?
not directly, but I am very close to that because I did my PhD in a group that was very much specialized in molecular dynamics. But I wanted to work with sequences and with this magic of from sequence to the structure thing.
All right.
In the end, I was right because it was before the language models, and then you see this happened, and I was like, okay, maybe I chose the right direction. I am currently collaborating, like in my group, there is a guy that is an expert in molecular dynamics, and I have to say it's very, very useful. It doesn't, let's say, excite me so much because, yeah, there is not much coding to do, let's say. It's more like tuning your parameters, tuning your simulation. Well, people likes it, but I don't. So I would love to like use these trajectories to let a model learn something about the force fields maybe, or yeah.
Is not there always an unknown quantum dynamics effect that is not considered in the thing that?
Well, there are a lot of yeah, it could be, but you don't have to go that far. I mean,
I know. Okay.
so proteins live in water, and yeah, so the molecular dynamic. So you need a force field for water. Imagine
What?
like a force field, like and you need to model water, let's say. So you have a lot. You have your protein, and you want to make your simulation like you want to simulate reality. So you want to simulate water. The point is that you have so many water molecules around, so you cannot do that at the atomic level for sure. Like you want your protein with all its atoms, okay, but the water around it, you can just do it. So people use models for that, and these models are not perfect, let's say.
I see.
And also for some systems you have to, so for some proteins you have to use a certain water model, for others another water model. I mean, so you don't need to go to quantum effects to see the limitations, let's say.
I see.
And it's also the computational cost can be reduced to do some approximations on
Yeah.
some atoms that are not that important.
I mean, in principle, you could put all the water atoms there, but
You have to start with water atoms.
it's like unfeasible. Like if you have, I don't know, 100 years to wait.
Wow.
No, I'm just making this up, but it's it's not doable, I think. Yeah. And in fact, yeah.
Sorry, now, it's just something I interrupted with, but I wanted to ask, what's your favorite part about your job and your research that you do?
What's my favorite part of what?
About your job and the research that you do?
You look fine.
Writing grants. Oh yeah. What I like. So what I really like is to solve problems by coding, like I'm a programmer. That's let's say to be very very general. Let's say. So that's the first thing. Then I'm really interested into these topics that I told you about. No, the best part is the freedom for me because I'm in a position in which I can basically choose what to research, which is quite common in research actually. I mean, yeah, sometimes like you are in a particular position in which you have to research those kind of things, but typically are the ones that you like. You're not there by chance, let's say. But maybe you are restricted to those. Sometimes I can pretty much tools, and that's very nice. I suggest you guys, let's say, aim to that. The independence in research is very important.